Good morning, everyone. Today's topic of discussion is Chapter 5 Cell Structure and Organization, Lecture number 4. So, the organelle which we are going to discuss today is mitochondria. Mitochondria is present in plant and animal cell. Singularly, we are going to call it as a mitochondrion. These are important cell organelles which involve in aerobic respiration. And mitochondria are absent in prokaryotic cell and red blood cells. Here the shape of this mitochondria can vary. It may be oval or it may be spherical or maybe sometimes spiral stripe like. Depends upon the cell in which it is present. We can also call it as species specific. Shape will vary. It is a double membrane bound organelle, and here the diagram shows this double membrane, the outer one called as outer membrane and the inner one called as the inner membrane. In the inner membrane we see that there is an infolding made with the same membrane and which is forming a finger-like projections called as a cisternae. Here the outer membrane is permeable to various metabolites due to the presence of porins named channels which will allow the entry and exit of the metabolites. But the inner membrane is selectively permeable and will allow only few substances to entry and exit. Now both the membranes are separated by a space. Inner membrane having the finger-like projections, it bears number of particles. These particles are called as oxysome. They can be also called as a cytochromes and electron carriers. So when electron chain system occurs, at that time electrons will pass from one electron acceptor to another. So these oxysomes are going to help and cytochrome are going to help for this purpose. Inner membrane is enclosing a cavity inside which there is a fluid matrix present. That fluid matrix contains circular DNA. So, up till now we have been studying that DNA is only present in nucleus, but yes, DNA is also present in mitochondria and these are called as a mitochondrial DNA. RNA is also present in the matrix of mitochondria and some types of ribosomes like 70S type. Various enzymes are present to carry out some pathways, for example, Krebs cycle, which are useful for the process of respiration. So the inner membrane which we have been talking, that inner membrane containing that oxysome, we can see the diagram over here. It's also called as a F1 particle, but as we can see the elaborated diagram, this F1 particle itself is having three regions: the head region, the pedicel called as a stalk, and the base region. So this F1 particle, overall it can be called as an F1 particle, but usually the head part is called as an F1 particle and the base part is called as FO particle. This F1 particle is also called as Fernand's and Morin particle, elementary particle, or else mitochondrial particle. Each particle we can see is embedded in the inner membrane of mitochondria, forming those cisternae. Now here the head part of this particle is having an enzyme ATP synthetase, and the foot is going to help as a proton channel. So the proton pump is involved with the oxysome, and also the ATP synthesis is also done with the help of oxysome. Next organelle is plastid. This is a distinguishing organelle for plant and animal cell because plastids are usually found in plant cell. Like mitochondria, plastids are also double membrane, and they are also containing their own DNA, RNA, and ribosomes. But they are larger in size and can be observed under light microscope. Plastids are classified according to the pigment present in it. So they are divided into three. First is leukoplast, second is chloroplast, and the third one is chromoplast. So here, if I go sequentially, we have a flow chart showing that plastid is divided into like leuco, chromo, and chloroplast. So first we will discuss leukoplast. Here leuco means colorless. It is a colorless plastid which do not contain any pigment, but yes, it's having a different shapes and size, and it is meant for storage of nutrients. Leukoplast is divided into amyloplast which stores starch, elaioplast that store oils, and eleuroplast that stores protein. Next comes chromoplast which contain pigments like chromo means color. So obviously it's containing a pigment like carotene which is present in carrot, and xanthophyll is another pigment which is also present in green leafy vegetables, example spinach. So color which they impart is yellow, orange, and many more chromoplast pigments can also give red color to fruits and flowers. Now you must have understood why potato looks white in color and why some flowers or shoe flower petals are red in color. So plastid is the one interesting which imparts color, and hence it improves the beauty of the flower. Next, both leuco and amyloplast do not contain photosynthetic apparatus. Leukoplast and chromoplast is not containing photosynthetic apparatus. We find it in a chloroplast. So now we are going to discuss about chloroplast, which is again a distinguishing characteristics of an autotrophs where they can prepare their own food. So plant cell, algae, and few protists like euglena, they contain chloroplast, which is ribbon shaped in some of the blue-green algae like Spirogyra. It may be different in size, it may differ in number and shape, depends upon the cell in which it is found. In plants, it is green in color or may be found in the green region of the plant, mainly in the mesophile of the leaf. So, chloroplast is lens shape in the mesophile of the leaf of a plant. Mesophile is nothing but the region just below the epidermis. Sometimes these chloroplast can also be oval shaped, spherical, discoid, or ribbon like. So shape varies Cell may contain one large chloroplast, for example chlamydomonas, or it may be 20 to 40 chloroplast per cell in mesophile cell. So number can also vary. Chloroplast contain green pigments called as chlorophyll along with other enzymes which will help for the process of photosynthesis, making food for plant. Now when we compare the inner membrane of this chloroplast with the outer ones, we have a diagram in the further slide. Then the inner membrane is comparatively less permeable than the outer ones, as we have seen in the mitochondria. Inside the inner membrane, we can see this. There is a stack like arrangement like a coin. You can see it's called as a grana. Okay, and grana is covered by a membrane called as a thylakoid membrane. Inside the grana we can see there is a cavity called as a lumen, and in the outer side of this thylakoid we see there is a stroma present, and this stroma is having its own DNA, that is chloroplast DNA, and the ribosome as well. So here we are seeing that chloroplast is also having its own DNA, right? And the grana and the outer side stroma both are helping for the function of photosynthesis. In detail, photosynthesis can be studied in the next chapter, or in higher standard we can say. This is how the chloroplast is. Next comes ribosome. Ribosomes are protein. We have seen many a times we call 70S ribosome, 30S ribosome. These are the protein factories because ribosomes are ultimately helping for the synthesis of protein. Ribosomes were first discovered or observed as a dense particle under electron microscope by a scientist called as a Plastid in 1953. Ribosomes are made up of ribosomal RNA and protein. In eukaryotic cell, ribosomes are also present in mitochondria, plastid, as well in cytosol. Cytosol is nothing but the matrix which is present in the organelles. Ribosome in cytoplasm are either bound state or maybe in the free state. We have seen many a times ribosomes attached to the endoplasmic reticulum and even the nuclear membrane. The attached one and the free state both are similar structurally and are of ATS type. Each ribosome is made up of two subunits, the larger one and the smaller one. Both bounded ribosomes are generally producing proteins which are transported outside the cell after the processing in ER and sent to the Golgi bodies. And from Golgi bodies, those protein can be sent to the targeted organelle. Further, the bound ribosome of acinar cell of pancreas are producing pancreatic digestive enzymes which will be used for digestion process. And the free ribosomes coming together and forming a chain called as a polyribosome for protein synthesis. So this is about ribosome. Thank you very much.